So today we are going to talk about nucleic acids. Last time, which is in module 3, we talked about lipids. And lipids is a topic in which you can see a lot of different types of structures and structures which will lead to different kind of functions, of course. So we talked about storage lipids. We talked about structural lipids. We also talked about the functions that some of these lipids have and how they can be really important. And as you finish that now, we are going to now move over to module 4, where we will learn different types of nucleic acids and what is the role of nucleic acids, like in terms of what is their role in making sure that cellular information is stored properly, and what will happen if for any reason they are damaged in any possible way. Okay, so this is what we are going to learn in module 4. And we are going to begin with some definitions, like always. So I would say that let us focus on just give me one moment, and let's just focus on some of the definitions. So we'll just start with this. So nucleic acids are large biomolecules. They are present in every form of life. Okay, so if we are talking of bacteria, viruses, yeast, trees, plants, different kinds of animals, everything has nucleic acids. So why does everything has nucleic acid? Is because there is a need to store the information of the cell. And that's exactly what the nucleic acid do. They store the information and they also pass it on from one generation to another. So I know some of you have done it, so you probably already know it. So let me just ask in general to try to find out how the class feels about it. What do you think? What is the most common nucleic acid which is used for storing information and passing it on from generation to generation? Okay, and you can also unmute yourself, like I said. At any point of time, you can ask questions. I just wanted to remind you guys, no question is small or big. Just go ahead, ask the question. I would rather really appreciate that than feeling like, wow, I got a little lost in last slide. Okay. So yes, that is correct. DNA is the molecule which is essential for storing information. However, there are some exceptions to it. And we do know that some viruses store their information in the form of RNA. Okay, so I want to give you a minute and, just take in this information. And let's also look at what is the monomer for nucleic acids. So what is a monomer? So let's try to remember what we studied in module 2. So when we're talking about carbohydrates, the monomers were monosaccharide. So in this case, we are going to be talking about nucleotides. So if it's a new name, it's not that complicated. We're going to learn a lot more about nucleotides as we go in some of the next slides. So nucleotides are monomers that make up nucleic acids. They are like DNA or RNA. Okay, so they are connected together to form the polymer. So now we're going to switch our attention to the right side of the slide to the figure. And on the figure, you see the structure of nucleic acid. And let me get, that's correct. Yes, perfect. So that's a good definition, Unedu. So we have here nucleic acid structure. So if you see here, the thing is that these are repeats. So if you see here, what you see over here, and then below that, and then below that, and below that. So it's a repetition. And so each of this unit is a nucleotide, and it combines together to form that structure. So we are going to take a look at what is each of these containing. Is this something that is familiar? And I'm going to tell you from now that there will be something familiar. So you are going to be finding a connection with module 2. Okay. So what does a nucleotide contain? A nucleotide contains 3 things. Okay. So there is a nitrogenous base. We're going to talk about it in the end. There is also a pentose and there is a phosphate. So let me just start with something that you are really familiar with, and that is the pentose. This is a sugar, actually. Just like you were studying about glucose, I hope you remember you also studied about ribose. It is a 5-carbon sugar, and that's what is shown over here. And I do know that all of you are familiar with the cyclic structure now, which is the Howard structure, and that's what is represented here. Now let us see what is this sugar connected to. So we are familiar with the sugar. Let us see what else is there that makes up a nucleotide. So what is here? Let's look on this side, which is much more easier because it's just a one group, and that is the phosphate group. That is a phosphorus connected to 4 oxygen. Okay, so I'm going to just try to see if I can do some highlights for you. So this is the phosphate group. And that was the sugar, which is shown in that box. And to your right, which is right here, is the nitrogenous base, which is the purine or pyrimidine base. And together, these 3 will make up a nucleotide. In the beginning, if you have never studied this, this may seem a little bit complex. Well, it's good to know that I'm not going to ask you to draw a structure, but what I really need you to do is understand that there are 3 parts, because all the 3 parts are really important to our understanding in the subsequent slides of what DNA is like, what RNA is like. So this is number 1, nucleotide, all 3 components. Sometimes while studying, you might also come across the word nucleoside. What is a nucleoside? A nucleoside is without the phosphate group. So that's what is missing. And finally, the nitrogenous base is also sometimes referred to as nucleobase. Now, in general, you will only be looking at nucleotide, but I do want you to be familiar with these terms just in case you come across something and I don't want it to be unfamiliar for you. So there's a lot to take in this slide. So I want to just pause and ask if this is something that you would like me to repeat, or if this was clear to you, the 3 parts of a nucleotide. Anyone has any questions about this particular slide?
Hey, Dr. M. I'm not familiar with this. And I'll just never mind. I'll just go back on the recording later.
Do you want me to tell this one more time for you so that it becomes like something,
if that's okay?
Yes, please. Of course.
Oh,
yeah. Happy
thank
to.
you.
Yes, I'm happy to go over it one more time. That's why I asked, because if I was doing this for the first time, I would be like, wow, that's a lot of structure and a lot of words. Okay, so I completely agree with you. So we are going to start with something familiar, which is the pentose, which is actually, and I'm going to write it down for you so it becomes something that you can relate to, and that would be ribose. And you will see how this term will quickly make a lot of sense, because you're going to look at some words which would contain ribose, and I'll ask you to remember that we talked about this. So this is the sugar. So the reason we are familiar with sugar is we did about carbohydrates already. So we are familiar with this concept. What we are adding to this is 2 different things. And if you notice, both of them start with the P. One is a phosphate group, and a phosphate group is always a phosphorus with 4 different oxygen. Then there is another group, and that is the purine or the pyrimidine base. Now, I know that this is not something you might have read before, but I'll actually tell you what this contains and the tricks to remember. And I actually made up those tricks, and I think those work out really, really well. It has worked out very well for the past students. So I'm pretty sure it'll work out for you. So all you have to remember is this pentose, which is actually a ribose with a phosphate group and a nucleotide base. So I'm going to write also here the nucleotide base. So you feel like, okay, this is something that it's not like someone is writing something and saying something else, because that is, sorry, let me write here the nitrogenous base. So because if I use different terms, nitrogenous base. So there is sugar, there is phosphate, there is nitrogenous base. To be really honest with you, this is like the reason this is my favorite topic is because this is the reason why we function the way we are. To understand like how life works, we need to really understand how DNA works, how RNA works. And this module will really help you in understanding. And trust me, we are going to come back to this structure. For now, we're just going to move a little bit forward and come back to this as needed. Okay. So we talked about those nitrogenous base, or just let us call them base for now. Okay. So there can be two types of bases, which is pyrimidine or purine. Pyrimidine has long name, but one, um, you know, one cyclic structure, and purine has a short name, but two. So that's how I remember. It's like pyrimidine is the one which is the smaller one, and purine is the one which is the bigger one. So what do these contain? These are, first of all, these are having nitrogen in them. So that is why they are called, if you remember, they're called nitrogenous base. Why do we call them nitrogenous? Because they actually contain nitrogen. Let's go back and see. This doesn't contain nitrogen, neither does this contain nitrogen. So it's nice to call that nitrogenous base because we would remember that this has N in it. And you can see the same thing over here. Okay, so it is aromatic molecule. And if you see here, if you remember from our nomenclature, these are the double bonds. Okay, so right here, you can see some of the double bonds. These are mostly planar structure, which means they are on like if you were to put them on a paper, they would be there. It's not like methane, which is like sticking out everywhere in a three-dimensional structure. You do not really need to remember these details because that's not something that we would be working on too much. Also, it's nice to know that they absorb light around certain region, because later when we are doing experiments with them, or maybe you are one of your guys going to go and do some research in a lab, or look at something, and maybe then you would need to know that it absorbs at a certain range. So it's nice to look at, be able to have that information handy. So don't worry if this feels like never seen the structure before, we are going to come back to it again. So let us now talk about the different types of nucleic acids. So nucleic acids are linear. Linear is just how carbon chain is, that it's one line. Polymers, poly is many, and polymers consisting of what? Sugars, phosphate, and base. So sugars is what I told you, the ribose, and then phosphate is the PO4, and base is what you were looking at just 1 minute ago, right here. So what are the two types of nucleic acids? One of them is DNA, and the other is RNA. So we are going to look at the full form, but before we look at that, we can see that they are differing by D and R. So what is D and what is R? So DNA contains the sugar deoxyribose, and RNA contains the sugar ribose. So you can see the difference between them. And what I have for you is the structures. Now, what you were to find out and tell me is that, and you have to assume that the numbering starts from here. If you remember, this is the anomeric carbon. We talked about anomeric carbon a lot of times, a carbon that is connected to 2 oxygen. So if this is 1, could you please point out what is the difference between ribose and deoxy? Why the name deoxy? What happened? Why it became deoxy? So anything that you can share with me about that?
Is it because there's two carbons?
Is it because they're 2 carbon? I will consider that as a thing. Let us see. I think there are other people writing things. Let's look at their responses as well. So I think someone wrote there is lack of oxygen. I like that. That's very simply and nicely put, Crystal. And also I see another response, deoxyribose has one oxygen less than ribose. Oh, okay. So yes, that is also another one, deoxygenated. D is removal. Okay, so try to think um, of some English word where D would become like less. Okay, so if you see here, there is OH over here, but there is only H here, which means that this oxygen, which was present in ribose, has been removed from here. And that makes it difference. That makes it different. So now you can see that that difference, does it really mean something? It means a lot of difference, okay? This really makes it a huge difference in their structure and also in their stability, okay? So we are going to see how that happens, but let us take it one step forward. So did everyone understand why or how ribose is different from deoxyribose? Okay, I'm assuming you did, but you feel free to ask question, if you did not. Okay, great. Thank you. So DNA and RNA are the most important molecules, in terms of cellular function. They are responsible for storage and reading of genetic information of all forms of life. Okay, so DNA is the one that provides the code for cells' activities. The full form for DNA is deoxyribonucleic acid. So I would like you to, at this time, pay attention to these words, which is the deoxyribo. So we talked about that, deoxyribose, and nucleic acid is the term that we did before, and ribonucleic acid. So remember that I told you that these sugar names will come up later. So deoxyribonucleic acid and ribonucleic acid. So one of them is important for storing all the information and passing on. The other one, which is RNA, converts the code into proteins to carry out cellular functions. So that is the role of these two. So again, this one is what information is stored in your cell. And this is what translates that information to something useful. Okay, so this is a complicated slide. So before that, let's have some fun. So let me ask you a question. So you have about 60 trillion cells in your body, 60 to 100 trillion cells. Does every cell contain the same DNA? Think about it. And if you can just answer in a yes or no, or you can also answer in a thumbs up for yes. I do think there are more reactions if you have downloaded the right Zoom. But could you share that my question to you is, you have 100 trillion cells in your 60 to 100 trillion cells in your body. My question is, do all the cells in your body contain the exact same DNA? Okay, so I see some answer, I see some yes, I see some no's. Okay, it's good to have some variety, I don't know is a great answer too, because, we really don't know. Now, what can confuse you is the fact that DNA actually is, every cell in the body is different. If not every cell, many cells in the body are different. For example, the brain cells are different from muscle cells and so on and so forth, right? So my question to you is, if the cells are different, I think that's what makes you think the DNA is different. But now I would like to remind you of the origin of any organism. Every organism starts from a single cell, and that cell is duplicated, and then it multiplies further to create all the organism. So remember, we all came from just one cell. if we all came from one cell, I'm now reframing the question, if you all came from one cell, and I just told you the DNA is like all duplicated and, it divides when the cell divides. Is it logical to think that the DNA in all the cells should be the same because it came from one cell? Okay. Some cells are haploid, some are diploid. True, but you did come from one cell. And that's what cell was the combination of, from the parental generation, right? So you did come from one cell. So, which means that if the same cell was the one dividing, then all the DNA in all the cells, the answer is everything is exactly identical. You can take out the muscle cells and genotype and you will get the same DNA. You can take the skin cells, same DNA. You can take any cells and you would find the same DNA, okay? So now I am going to go to the other part of the mystery. If DNA is same in all, right? If DNA is same in all the cells, what makes it different? Is it have you heard of proteins? So is the proteins different in different cells? So I hope you're understanding my question. So my question is something really important, is that all the cells in your body contain the exact same copy of DNA. Why are they different? Any thoughts? No thoughts, really? Okay. Oh, no. Good thinking. I appreciate the thinking, because RNA is different in different cells and the proteins are different in different cells. So all the cells have the same exact copy of, yes, the same exact copy of DNA. Only a part of it is used to make RNA, and that converts to protein. And so if you have the same cell, but your eye cell looks different from muscle cell just because the proteins are different. Okay. So I want you to early on think about the process, like what makes you such a unique person, each person having so many different cells and tissues. So let's come back to this topic. So deoxyribonucleic acid. So we said there is a sugar. We always start with the sugar because that's what we have studied before. And then we have the phosphate group. And then we have the base. So these are the three things that comprise of that make up a deoxyribonucleotide. Okay, so those are the three things. Now, what about ribonucleotide? We know it's going to look very similar. A ribonucleotide will have a ribose with a phosphate and a base. So it's the exact same thing, except that this has oxygen here, where that other one did not. It was deoxygenated. Okay, so I hope that difference is a little bit more clear. Okay. So now let's go back and see the slides that we missed. So what about these bases? Like, what are these bases? So we talked about that there is purines and pyrimidines. So purines there are two types of purines and two types of pyrimidines. Okay, so let's first look at their names. So first of all, there is adenine and guanine. Adenine and guanine are purines. And then there is cytosine and thymine, and those are pyrimidines. Now, if you are seeing this for the first time, it may be a little overwhelming. So I'm going to, like I said, I'm going to give you a little trick to remember this. And my little trick to remember this is, look at that. This is CY, cytosine, and this is THY, thymine, and this is PY, pyrimidine. So the pyrimidine is the one that has cytosine and thymine, okay? Whereas the guanine and adenine do not contain the Y. So purine can be either guanine or adenine, which is the double aromatic rings. And pyrimidine is the one that is single aromatic ring, and that is either cytosine or thymine. So these are the 4 different bases that are present. I do want to pause here and ask if you have any questions from this slide or the previous slide. Anything
I'm
at all?
fine. I'm just trying to keep up. That's all.
Okay, that's good. That's good. Yeah, that's good news. You know, we all want to keep up with that, because, you know, who wants to just start all over again just because you missed something? We don't, no one wants that. I wouldn't want that. Okay, so if you see here, there is, let's see. So what happens with these bases? What do they do? Like, why do we talk about these? Why have we been talking about these bases? Why are they important? They are important because they are present over here. So now for a minute, let me just try to make this disappear from your side. So I'm going to remove these so you don't like get distracted, and remove this. So this is how a single molecule looks like: the sugar, the phosphate, and the base. Well, that's nice to know. Also, there can be different types. You know, a thymine would have only one aromatic ring, whereas a guanine has 2 aromatic rings. And similarly, an adenine and cytosine is different. Now look at the magic. The magical part here is these bases actually bond with each other through a hydrogen bond. So if you see here, between oxygen and hydrogen, there is a bond formed. And similarly between nitrogen and hydrogen. So you can see that this is forming 2 bonds. And this guanine and cytosine is actually able to form 3 bonds. So I think it's like common logic to think like 2 bonds are weaker than 3 bonds. So this must be a stronger bond between cytosine and guanine. So the reason we are studying all this is to say that the bases actually will form bonds. Now let me show you where that bond is. Okay, so that bond is, oops, right here. If you see the structure of DNA, I'm sure everyone has heard that DNA has a double helical or a double helix structure, double because there are 2 strands, and helix because that's how it looks. So what keeps this helix together is this hydrogen bond. This is the one that is keeping the helix together, this hydrogen bond right here. If this hydrogen bond was not here, this would not be a double-stranded structure because this is what is keeping it together. So that's what is indicated by these blue arrows. Now let me show you one more interesting thing, and that would be, look at this. So look, these sugars are combined together through this phosphate bond because one of the phosphate molecule is holding a O, which goes to sugar 1, and the other O goes to sugar 2. So this together is also getting connected. So where is this? So let me show you where that is. So that is actually right here. I'm trying to draw it in red so you can see. So this is these connections between sugar, that's right here. So sugars are doing this red part that is making the backbone of the helix, and the middle is being made by this. So this is the hydrogen bond. So I'm just trying to, it's not a perfect arrow. So excuse me for my horrendous drawing because of this kind of tools Zoom has. But these hydrogen bonds in the middle, okay, that's what it is. And these phosphate bonds in the end. I want to pause here, ask you if this was clear or if there are any questions about this part.
Where's the arrow pointing for the hydrogen bond?
Okay, so let me make a box. Box probably is going to be more clear. And actually, let me maybe erase this. So the hydrogen bond is like, this is hydrogen bond, this is hydrogen bond, And that's what is in the middle, like sorry, too big. This is all hydrogen bond. You see the yellow and red being connected and the green and blue being connected. That's where this connection is, because the hydrogen bonds are the one that is keeping them connected.
Okay.
Okay. So one part is holding the center, one part is holding the back. That is what makes it. So one part is holding the back, and that's what makes this double helix together. Now, in reality, these bases don't have color. Like if you were to actually be able to see them, they have no colors, but just to make it convenient, adenine is shown by red, thymine by yellow, guanine by blue, and cytosine by green. Now, if I want to remind you, this adenine, thymine, guanine, and cytosine were on the previous slide. Okay. Okay, so let's try to, let me erase this. Let me clear all of this. And draw your attention to one last thing before we move on. And that would be, look at these numbers. So carbon, like I said, mentioned, anomeric carbon starts it. So 1, 2, you know, these corners are nothing but carbon, 3, 4, and there is one more here, 5. So that is what the numbering is. So 5 is the carbon. That's what's number shown here, is where the structure starts. And where does it end? It ends at 3, because one is the top part and the other one is the bottom part. Before we leave this page, I want you to note one thing, and I'm going to show you something, and you're going to just tell me if it is parallel or anti-parallel, okay? So I'm going to show you this sugar versus this sugar. Look at the O and tell me if they are parallel, which means that they are in the same direction and like almost like parallel lines, or they're anti-parallel, which means they're parallel to each other, but they're not facing the same direction. So look at it carefully and tell me parallel or anti-parallel, and you can just say A or P, and that would be just fine. And you can also say, I don't know. Anti, okay. They are opposite to each other. You can see this oxygen is facing up. This oxygen is facing down, okay. So what we have here in nutshell is this structure, which has this sugar phosphate backbone. By backbone, we mean the light blue ribbons. And we also have these bases, which make the hydrogen, which is what keeps this together. So this is what is really important for you to know, like structurally or structure-wise, this is what it's important for you to know. Now about RNA, we talked about this particular slide because I showed you the ribose sugar is right here and it has an oxygen. So that's what is different. You can see that this did not have the oxygen, and in ribose, it does have the oxygen. And RNA also has the same basis, exact same basis. But there is one difference. There is still guanine and adenine for purines, and there is cytosine, but the thymine is gone. In place of thymine, it's replaced by a uracil. So that's something that you need to know. Now you might ask me, do I need to remember that the 4 words adenine, guanine, cytosine, thymine, and additionally uracil? So that makes it 5. Do I have to remember this? And I would say, yes, absolutely. Is this something that can be asked in the exam? Answer to that is also a yes. Okay. So these 4 terms, maybe you want to write it down. It's not that hard. Once it's kind of rhyming, so it's not bad. You can remember. And like I told you, the Y ones are the one that is, uracil is an exception, but the one containing Y, cytosine, thymine, and uracil. Now, RNA is single-stranded. It's not double-stranded. It just cannot form the bond. It's not able to form the hydrogen bond just because of the way its structure is. The presence of an extra oxygen prevents the formation of a bond. And these are the 4 bases. Now I'm going to pause here and ask you an interesting question, which you're allowed to Google and tell me. We've been hearing about this coronavirus all this time, like coronavirus, right? What kind of nucleotides nucleic acids does coronavirus have? Is it DNA or RNA? And you're looking at SARS-CoV-2, okay? So thinking about the SARS-CoV-2, so look it up and tell me, and sometimes RNA is even double-stranded. So what kind of RNA is it? If it is an RNA, is it a DNA? What is it? So take your time, look up what is the genetic material in coronavirus. Yep, and it's kind of tricky when it's RNA. It's kind of tricky. It is, it's a little bit, different mechanism by which an RNA virus would start making proteins. So it is an RNA virus. So it's not a DNA virus. We are having DNA as the genetic material. The SARS-CoV-2 virus, which causes COVID, is the one that contains RNA, and it is a single-stranded RNA. Okay, so let's review. Now we are, I guess, in a little bit frame of mind to see all these just structures. So we do want to take a look at these again and again, not because they're complicated, but because we need to memorize them. So the purines are the ones that are adenine and guanine. They do not contain any Ys. Pyrimidines are the ones that are cytosine and thymine. That's where the Y is. And thymine and uracil are so close to each other. Look at their structures. Wow, they're like, what is the difference between their structure? Let me see how well you can articulate that. What is the difference between thymine, which is present in DNA, versus uracil that is present in RNA? Yes, that methyl group that is sticking on C5, that's present in thymine, but not in uracil, and does make a lot of difference. Okay. So let us try to put all of this knowledge of chemistry and structure together. The DNA is a very important molecule in all living organism. Any of this, especially eukaryotic cells, there are 2 kinds of cells. There is eukaryote that contains a nucleus, and prokaryote that does not contain a nucleus. It's a little bit more simple. Inside the nucleus, if we just blow this up, a chromosome, that's how it will look like. If you blow a part of the chromosome up, it will look like this, okay? And so, and if you were to unwind this was going to look, actually then start looking like a DNA. And some chunks of DNA make the gene, and the other chunks don't make anything. Okay, so some of them make the gene and the other ones do not make anything. So that's something that we also need to know. So in all your 60 trillion cells, there is nucleus. Inside the nucleus is the DNA, and that DNA is actually bound together and put in as chromosomes. And this is all happening in a space of, I'm trying to think, I think it's 0.05 millimeter. It's like really, really small. Okay, it's really small. Okay, I'm going to come back to this slide. Okay, I cannot go that far. Okay, so then this DNA in most of the cell is diploid. Okay, in some plants there are many copies. So which means the diploid means 2 identical copies. So haploid is 1 copy, diploid is 2 copies, and polyploid is multiple copies. Okay, so let's talk about diploid. So all human beings have 2 identical copies of DNA. One comes from the mother and one comes from the father. Okay. What about polyploid? Does anything that we know have multiple copies of chromosome? Yes, strawberry has 8 copies. Each cell has 8 copies of DNA. Why is that? We don't know. I mean, we just know that it's there. If the haploid number, so haploid number is half of diploid. So if the haploid number in a human cell is 23, what is the diploid number? Diploid is haploid times 2. 23 times 2, that is 46. So now there is a question for you on the bottom. If the diploid number of an organism is 50, so mark that this is diploid. What would be the haploid? What is, by that I mean, what would be the half of this?
25.
25, yes, perfect. So that's another question. Is this important, guys? This is important for your exam. Okay, so I hope that at least you can understand the concept of haploid and diploid. So haploid is literally the N, and this one is 2N. So that's the double, okay? Okay, so when we talk about RNA, so DNA is pretty simple. There is only one kind of DNA. But when we talk about RNA, there are different kinds of RNA. We will review them next time as well, because at this point, like, you guys have taken in so much information that this may seem like, wow, this is actually a lot, right? So I just want you to know that we will be repeating this one more time. So there is messenger RNA, which is the one that is responsible for converting the information from DNA to protein. There is ribosomal RNA, which make up the ribosome. And there is transfer RNA, which helps in making the protein by bringing in what we call an amino acid. And that's what is the building block of protein. Okay, so we come to the part where we talk about how DNA is converted to RNA. So DNA is the one that contains all this information. It is converted by a process called transcription to RNA. And that RNA is then converted, which is this red one, is then converted by translation to a protein. So DNA to RNA transcription, RNA to protein is translation. So transcription, remember DNA was inside the nucleus. So all the conversion of DNA to RNA, that is transcription, is taking place inside the nucleus. Whereas the RNA to protein conversion, we need the protein outside. So it's happening outside the nucleus. Okay, so what we're going to do is we're going to do this interesting exercise, which you will need to do the assignment. So for those people who would like to finish the assignment in advance, this would be something extremely useful. And then we may be able to pause because, like I said, lots of information for today. So I just want to make sure that we leave it at a point which will help you do the quizzes or assignment. So transcription, like we said, converts DNA to RNA. How does that happen? That's what we're going to look. And then translation converts RNA to protein. So first of all, when we see these big words, we're like, how are we going to remember this? Because I might write an exam that RNA to protein is transcription and DNA to RNA is translation. So I have a tip for you. Arrange this alphabetically. What comes first, transcription or translation? So we know that C comes before L. This comes first. So that's the first process, DNA to RNA. The second process is the one that comes later, RNA to protein. So transcription is DNA to RNA. RNA to protein is translation. Now, for this to happen, we need to look at this particular slide carefully. And this is a representation of a double-stranded DNA. And if you see here, adenine is paired with thymine. So I'm going to write down here. Actually, I should have a separate slide for this. Adenine pairs with thymine. So in other words, I'm saying A with T. Okay. And cytosine pairs with guanine. So for those who are thinking about uracil, remember uracil is not in DNA, it's only in RNA. So we are only talking about DNA. Adenine pairs with thymine, so A with T and C with G. So look on the top. This is the sequence of the DNA. That's what it looks like. You know, when you are writing it down, that's what it looks like. And then this T pairs with A pairs with T. It doesn't matter what pairs with what. It's always the same. And then, yes, and then C pairs with G or G pairs with C. That's how it is. That's how it is done. Okay. So if we look here, there is this what we call as complementary bases. So this T is complementary to A is complementary to D. You can even see that the little thing to shown here that they just kind of complement each other. A with T, A with C. So what happens if by mistake A pairs with C? That would be a mistake. That mistake is called a mutation. Can that happen? Of course, because DNA is a is something. DNA replication is happening in nature. Nature can make mistakes. So A can pair with C, but if that happens ever, that would be a mistake. So that's why T must pair with A and C must pair with G. And if you see here, so I'm going to ask you to focus your attention to just this part and compare with this part, and you can see that all the T, let me show with another thing, all the T has been replaced with the U. Wherever there was a T, in RNA, what has taken its place is a U. We're not going to worry about this at this time, but I'm just saying T is made as a U when it comes to RNA, and it's not double-stranded, it's just one, which is the bottom strand that is happening. So this is the starting point, which is called start, and this is the ending point, which is stop. And what is a codon is a 3-letter word. So we are going to talk about codon in a little bit. We're not going to rush into that. Let me clear this. I want to know if you have any questions from this slide, and if you were unable to correlate it with the previous slide, then if you can ask me specific questions, that would not only help you, but also might help other people in the class. Okay, you understand this? Okay, fine. If you understood this, this is what we are doing. You are going to do this for me. You make a complementary sequence for this one and write it down in the chat. So what you need to do is you need to tell me what comes here, what comes here. You can just write like I wrote continuously. Oops. Copy, look at it. Look at the rule. The rule is that A pairs with T and G pairs with C. Look at that rule and make this bottom part, considering that this is 2 strands of a DNA. Strand 1, strand 2. So fill that up. This probably is going to be one of your assignments. So I hope that you are able to do this. So I don't know if what you need to say is correct, or Taylor said, so I would like more people to respond. Okay, good. I see a lot more responses, and I'm totally okay with you writing in smaller or lower case. I hope that more people can respond to this one. What does U pair with? Good question. U pairs with A. Actually, should have written this down here.
Question.
T,
So you will
yes.
only pair with A for RNA, and then DNA, A pairs with T.
That's correct. Absolutely. Okay. Absolutely. So if you are not getting this for any reason, you should ask. You can ask now, you can ask later, but this is what it is supposed to be like. Okay. And like I said, we will be doing this kind of transcription again. So here we go with a learning check, and you can do this by yourself. I would say that if I would be you, I would go through this seriously and go back and forth and see if I'm understanding. So one thing that I noticed in oral exam is that sometimes students would go over a concept and think this is not difficult. I know this, but when you start doing question, it feels like a little bit tricky. The question feels tricky. So I think one of that is like you should try to go back and do it again, or do some of the questions like here and try to answer them and not skip this and try to do this. So in a nutshell, let's combine together the differences between DNA and RNA. DNA is double-stranded, D for DNA, D for double-stranded. RNA is generally single-stranded. It can be double-stranded, but generally it is single. DNA has deoxyribose sugar, which does not have this oxygen, deoxygenated. And RNA has ribose as the sugar, which does have the oxygen. So please note that difference. Bases used. Both of them use adenine, guanine, cytosine. DNA uses thymine and RNA uses uracil. And there is a difference between those two bases because uracil does not have the CH3 or the methyl group. So does anyone, when you are looking at this summary, does anything look unfamiliar? And if that is so, then you can ask now. And if someone is having an exam with me, we'll just do it like right after we finish this. So. Okay, so I hope that everyone is understanding. So what we have done so far is we have tried to learn what is DNA, what is RNA, what is common between them, what are the differences, and how can we convert one form to another. Yeah, we can stay here for the exam. Yeah, anyone who is doing exam can stay on the same link. That's fine. And if you did not schedule an oral exam, I mean, we can talk about it even now. That's fine. So we are going to go over a little bit a history of DNA next time. So we still have a little stuff to go. Let me show you how much we have remaining. So we will be talking about how the DNA was discovered. And again, we'll go over the rules of how DNA has A pairs with T and C pairs with G. We will talk about base pairing again, a little bit, just to make sure that we have understood this correctly, because sometimes when you write like this, it feels like very different from when you actually make that helix and write it like that. So we're just going to familiarize ourselves with those forms and how they're complementary to each other. Then we will start talking about how to convert that information. So we talked a little bit about transcription. We will also talk about how translation happens. These are very, very important. Remember, you guys are stepping into the allied health field at the time where a lot of it is becoming personalized medicine. So I'm not sure if you've heard this, but personalized medicine means that patient care is becoming more and more catered to individual, based on individuals, such as even individual genome, right? So as to understand the personalized medicine, the first step is to understand that every human being has a certain genomic DNA. And of course, that makes certain amount of protein. So understanding the profile of a patient and why is it different, you need to have this kind of basic knowledge, and that's what is going to tremendously help you. And if you're going to take a microbiology class, you might have already done this, or if you're going to take, you will be also doing that in this class. And so it will be beneficial to learn this. So we will be talking about translation, followed by some of the functions. And then the rest of the slides, which are more general, will be dedicated to like what happens when DNA gets damaged. Does it get damaged? What happens when it gets damaged? What is the effect? And is there anything good about radiation? Because we always associate radiation with something that is bad. But we do know that in the health field, radiation is an important form of therapy. And so that's what a little bit we are going to be focusing on too. So let me just stop sharing and ask you guys if, and so stop recording.